more or less slowly. First of all, the photoacoustic effect. The photoacoustic effect is generated when a variable light interacts with a strongly light-absorbing material. In water, the photoacoustic effect may produce hot bubbles and shock waves that could affect the integrity of nearby cellular membranes, opening transient pores. And this is what is called photoporation. The aim of this study was to evaluate the effect of pulsed laser irradiated carbon nanoparticles on model membranes or cell membranes. So we use carbon nanoparticles as the particles that will be heated up by the laser radiation. And these hot nanoparticles will induce the thermal waves that will modify, they will actually break down the membranes, both the membranes of cells and of the sort of vesicles that we call liposomes. We just had a very beautiful introduction to laser light by the previous speaker, Professor De La Fuente. So I don't need to go into much detail, just to tell you that a brilliant B pulsed neodymium YAG laser was used. The pulses were 6 nanosecond length at a frequency of 10 Hertz. Pulse energy varied between 0 and 56 millijoule per pulse, and wavelength of the emitted laser infrared light was 1064 nanometers. We have discussed the laser, now the nanoparticles. We used carbon black nanoparticles that we abbreviate as CNP, carbon nanoparticle, in suspension, and they were prepared. The nanoparticle, I mean the carbon black, was courtesy of Cabot Corporation in Belgium, and the nanoparticle suspensions were prepared in aqueous solutions of a non-ionic surfactant called poloxamer 407 by gentle sonication. In a bath sonicator, we let them for 15 minutes or so in a bath sonicator and with the detergent, and we obtained rather stable suspensions of carbon black nanoparticles. Now, the other element in our experiment, the liposomes. Liposomes, in general, are artificial vesicles formed more or less spontaneously when phospholipids are dispersed in water. Phospholipids are the main components of cell membranes. The main lipids in cell membranes are phospholipids. If we extract the lipid from the membranes and then disperse these phospholipids in water, we will get these spherical structures that we call liposomes. Spontaneously, we obtain the MLV or multilamellar vesicles. We can sonicate these multilamellar vesicles, and then we obtain the SUV sonicated small unilamellar vesicles. Or we can alternatively extrude these MLVs through polycarbonate filters of defined pore size, and we obtain what we call large unilamellar vesicles, LUV, between 100 to 150 nanometers. In our work, we used LUVs of about 100 nanometer diameter. Oops, sorry. This is the extrusion method. So here we have on top the multilamellar vesicles that can be microns in size. And here we have the polycarbonate membrane, in this case with 100 nanometer pores. And we do not understand very well, frankly, why this happens. This is a bit of black magic, but the fact is that if we put pressure so that we extrude these multilamellar vesicles across the membrane, we obtain much smaller 100 nanometer vesicles, unilamellar vesicles here. We can follow the process by light scattering, dynamic light scattering, so that we measure the sizes, and after we repeat this procedure, this extrusion, between 10 and 20 times. After a single time, we are already in the region of 200, 300, but by the 11th extrusion step, we have already vesicles that are 100 nanometer in size. This is what happens. On the left-hand side, we have the multilamellar liposomes, several microns in size. At the right-hand side, we see the unilamellar vesicles, about 100 nanometers in size. So this is what we are seeing. Oh, now I have a good pointer. Okay, okay. It will be better from now on. So now we have the same vesicles, but seen with freeze fracture electron microscopy. And these large unilamellar vesicles were composed of phosphatidylcholine, and phosphatidylcholine is the most abundant phospholipid in mammalian membranes. So we see them very homogeneous and very nice. With cryo electron microscopy, they look even more beautiful. And you see they are very homogeneous, very regular. And here we have in the same image a carbon nanoparticle and a large unilamellar vesicle. So they are both of the order of 100 nanometers. So we can see both. This is just an example, but I can show you some further examples. These are just the vesicles without carbon nanoparticles, and this is the mixture of nanoparticles and vesicles. And you can see relative sizes, and you can see how our preparation looks like. So first, I will tell you our studies with liposomes, and then we will go to studies with cells. When we prepare the liposomes, the large unilamellar vesicles, LUV, we can load them with whatever water-soluble substance we want. And in this particular case, we loaded the vesicles with a fluorescent water-soluble molecule called calcine. And we could see the leakage or flux of the entrapped calcine by measuring fluorescence lifetimes of calcine in vesicle suspensions prepared and treated in different ways. You have this reference here for details on the method to study leakage of calcine. So the idea is that at a high concentration, calcine fluorescence is self-quenched. So inside the liposome, fluorescence is very low. But when the liposome breaks down and calcine is released, then the fluorescence intensity increases because self-quenching does no longer occur. Not only that, not only the fluorescence intensity increases, but the fluorescence lifetime also changes. It is more convenient for us to measure fluorescence lifetimes than fluorescence intensity in this particular experiment. So, first of all, we did a calibration with different concentrations of free calcine that should correspond to different proportions of calcine release from the vesicles. The straight line is the theoretical line, and this is the calibration in blue triangles, and you can see that the calibration follows very well the theoretical line. Now, what we observe in general is a partial dye release, a partial efflux of dye. So at the end, the vesicles are neither totally full of calcine nor totally empty. There is a partial dye release. And this may originate either from a fraction of the vesicles being completely emptied while others remain intact. So we would have some vesicles totally empty and others intact. Well, on average, we would see partial dye release. And this is what we call the all or none mechanism. And it is caused by either permanent holes or slow closing holes. If we make a hole, a permanent hole, then all the dye will leave. Or if it is just a slow closing hole, if the hole is open for a long time, then everything will go out. So we will have damaged vesicles that are empty and intact vesicles. And the overall effect will be partial dye release. But another possibility is that the vesicles, all of them or most of the vesicles, release only a part of their content, and they keep the rest. So all of them are on average the same. They have released only part of the contents. And this happens if the openings are transient, and it opens and immediately it closes down again. So only part of the contents can release, can leave the vesicle. And this is what we call the graded mechanism. There is, fortunately, a good experimental method to distinguish between these mechanisms. And as I said, they can be distinguished by fluorescence lifetime measurements. So we measure the fluorescence lifetime of calcine because when it is entrapped and when there is a lot of self-quenching and the fluorescence intensity is low, then we have a lifetime, a short lifetime of the order of 0.4 nanoseconds. While in the case of the free calcine, when it has left the vesicle, when it gets diluted, then the fluorescence intensity increases. The lifetime of fluorescence of the free calcine increases by 1 order of magnitude, about 4 nanoseconds. And again, here I give you a reference, a paper in the Biophysical Journal 2015, where you can find the details of this procedure. So this is what we did, and we look now to this plot, to this figure, in which we have plotted the percent efflux with the lifetime of calcine. We have done 3 different experiments. In one of them, the blue triangles, we have a mixture of LUVs, of vesicles, and of free calcine. So the calcine is outside all the time because we have put them like that. We have added the vesicles and then the calcine, and you see that most of the time the efflux occurs at 0.4, and this is also what happens when we add Triton X100. With Triton X100, we have an all or none release of the vesicles. And this is the line, the perpendicular line, is the theoretical line for the all or none mechanism. With Triton X100, most of the time, we have all or none. When we add free calcine outside, of course, it is us having all or none. And this is what you see here. You have either vesicles containing calcine inside or empty, flat vesicles. However, this diagonal line indicates the graded release mechanism, in which you see some vesicles contain plenty of calcine, others less, other much less. And the black experimental points correspond precisely to our experiment of carbon nanoparticles and laser. So in our case, it is quite clear that our vesicles are being opened only transiently by the shock wave generated with the laser heated nanoparticles. And then the pore is immediately repaired. And this is why we do not have any really empty vesicles, but all of them contain a fraction of the original amount of calcine. We have this graded release. Just to repeat what we saw, when vesicles were irradiated with pulsed infrared laser light in the presence of carbon nanoparticles between 0 and 80 microgram mil, lifetime measurements showed a trend clearly closer to a graded than to an all or none mechanism. The result supports the idea that the photoacoustic effect may cause in membranes a transient physical disruption. And the disruption would be long enough to allow some efflux, but immediately it would be repaired to return to a stable integrity state. You see, again, I want to repeat, with Triton, we have most of the time all or none, except in some extreme cases. When we add LUV and free calcine, we have the equivalent of all or none, except here some calcine is getting inside the vesicle. But in our case, in this range of efflux between 0 and about 70%, it is all the time the graded mechanism. So now we go to our studies with cells. And we studied CHO, they're called Chinese hamster ovary cells. They're very commonly used in cell biology studies. They can be very conveniently grown and kept, so they are very popular. And they can be also suspended. They can be stored flat on a surface, but adhered to a surface, but they can also be suspended, and they are quite happy in suspension. So the photoacoustic effect was tested in real cells by studying the intracellular uptake of calcine from solutions when suspended CHO cells were irradiated with pulsed infrared light in the presence of carbon nanoparticles, exactly the same as we had done with liposomes. Only what we see is the uptake of calcine, because with liposomes, it is very easy to load them with calcine. But with cells, this is much more complicated. So we see the opposite effect. We see how, at least initially, we will see how calcine gets into the cells. And because we were worried, because cells are much more fragile than liposomes, we were worried that the laser nanoparticle treatment would kill the cells. Well, we treated them with propidium iodide, and propidium iodide is a stain that is selective for dead cells. So we look at the preparation with the confocal fluorescence microscope, and we can see the dead cells stained. If there are any, they would be stained with propidium iodide. So here at the left-hand side, we can see the overlapping of three images. We have seen this CHO cell suspension by dark field, and this tells us the contours of the cells. The contours of the cells are obtained with the dark field technique. Now the green color is the calcine fluorescence. So everything in green is fluorescence. And the propidium iodide that signals dead cells is the red dots. So we have only 1, 2, 3, 4, very few dead cells. All the others are alive, and most of them are loaded with calcine. So CHO cells treated with laser and carbon nanoparticles have taken up calcine. In this panel at the right-hand side, it's just the same, but only the green calcine is seen, so that you can observe that most cells contain calcine in different amounts, but most of them contain calcine. So we see that calcine has been delivered to the interior of the cell, and this has happened specifically by the photoacoustic effect, because we did some controls in which either we irradiated with laser, but we put no nanoparticles, or in a different control, we put the nanoparticles and we did not irradiate. Okay, in none of these cases did we see calcine uptake. You see here, left-hand side, irradiation, no carbon nanoparticles. Well, yes, we have one cell with some calcine. As usual, a very few dead cells. And in the other control, we did not irradiate, and we did put carbon nanoparticles. The amount of dead cells is even slower with no irradiation. There is one cell, one loaded cell here, but obviously nothing to do with what we had seen previously. Very important, cell survival is practically complete, at least in terms of immediate cell death and integrity, because only 1 or 2% of the cells appeared marked in red. Again, you can see that only 1–2%, even less here, of the cells appear marked in red. Red means a propidium iodide absorption, and this is a sign of cell death. Now, what is the mechanism? In liposomes, it was a graded mechanism. What is the mechanism in cell? Well, again, the process seems to be driven by a graded mechanism, as shown by the presence of cells with different inner concentrations of calcine, ranging from only residual calcine to strongly bright green cells. Let me show you this again. If we look at the image confocal fluorescence microscope of the cells loaded with calcine, we see some of them are very bright, so they have been stained. They have uptaken a lot of calcine. Others are very dimly stained, indicated that they have taken a small amount of calcine. So this is another example of a gradual or graded mechanism, only here it is a mechanism of uptake. In the liposomes that we saw in the LUVs, it was a graded mechanism of release. We studied different laser energies, what is called the fluence, the energy per square centimeter, 50, 100, 200 millijoules per square centimeter. And obviously the uptake of calcine increased with the laser energy. And this is I think as a function of time. I cannot see very well the bottom of the image, but I think it is. So the result of our studies of calcine release from Chinese hamster ovary cells treated with infrared laser and carbon nanoparticles is, sorry. Now we go to a different step, different experiment. And this is calcine release. So we took the previous cells, these cells that had been loaded with calcine, we cleaned them, we put fresh buffer, and then we add fresh carbon nanoparticles. These are the calcine-loaded cells. Then the samples we place under the confocal microscope, and simultaneously we irradiate with the femtosecond infrared laser at a wavelength of 1000 nanometers for 120 seconds. So what we obtain is a live recording of the photoacoustic effect producing transient openings in living cells, because we do it under a microscope. And in the same setup, we had the confocal microscope and the laser light irradiating the samples. So we could see live the photoacoustic effect. And I'll tell you about the videos later, but I'll show you now only the first and last frame of these videos. When we do not put carbon nanoparticles, we just irradiate with laser, then, well, essentially nothing happens. The laser by itself is not causing any cell breakdown or release of calcine. However, when we put laser and carbon nanoparticles, then we see that most of the calcine is released in 2 minutes, in 120 seconds. Most of the calcine has been released, so the cells that were here very bright are here very dim. We can quantify this, and we can measure the time course of cell fluorescence emission without carbon nanoparticles. These are the two top images, and with carbon nanoparticles, the two bottom images. So we can see, as we could with the naked eye, we can see quantitatively that in the absence of carbon nanoparticles, the cells do not lose any calcine. However, when carbon nanoparticles are present, we see this gradual release or gradual decrease of fluorescence emission, indicating that calcine is being lost from the cells, is being released from the cells. So, for quantification and standardization of calcine intracellular delivery and cell viability, we used flow cytometry because we wanted to measure simultaneously and precisely both magnitudes, intracellular delivery and cell viability. We wanted to make sure that our cells were alive when they were taken up calcium. Cell viability was determined not only by quantifying necrotic cells with the flow cytometer, stained in red with propidium iodide, but also we measured cells in early apoptosis by staining with an Xing5 label with the fluorescent marker FITC. So this is, we measure two different things. One is dead cells with propidium iodide and cells in early apoptosis, very likely to die, but with a totally different method, Xing5. And we believe that this is a more realistic approach to cell viability, because cells in early apoptosis should not be considered viable in the long term. Early apoptosis means virtually always death in the long term. So these are examples of flow cytometry measurements at different times. Here we measure calcium uptake. You see, calcium uptake at time 0. More and more calcium is located here, and less is located here. So this is a quantitative measurement of calcium uptake. Is not just looking at the cells. A quantitative measurement of calcium uptake, in this case at 60 millijoule per square centimeter, but we did, of course, this under the different conditions. At the same time, we were seeing viability. We were seeing viability by measuring propidium iodide cell death and annexing 5 early apoptosis. So we can see that at time 0, the cells are alive and well, but as soon as irradiation starts, these areas are getting populated. So the number of cells in apoptosis is increasing, and the number of cells with high propidium iodide increases. So we end up with cells with high propidium iodide and also starting the apoptosis procedure. So we have, with these results, we can decide what are the best conditions for intracellular delivery. We want conditions under which the cells take up the calcium, but they do not die in the process. So we have selected at a fluence of 60 millijoule per square centimeter. We could find a point in which uptake reached 80% of the cells. Cell death did not exceed 10%. And the most appropriate time for that was of 40 seconds. We have this, first of all, we have this plot at the left-hand side. After 120 seconds, we measure calcium loading and cell death. So obviously we want to be more or less in this region. And this is as a function of time. And as a function of time, we see that after 40 seconds, we have almost maximal calcium loading and relatively low, about 10% cell death. So these are the conditions that we would recommend for loading cells with calcium or whatever other reagent that we fancy for loading cells with the laser carbon nanoparticle photoacoustic procedure. We not only did that, we also looked at long-term viability by reseeding the cells on a flat surface and growing them for 48 hours to verify the maintenance of the properties of a native healthy cell, that they attach to a surface, they spread, and they divide. So with no laser, we see plenty of cells there attached and very few released. With 46 millijoule square centimeter, we also see plenty of cells there, but they are more rounded. And with 66, we see less cells, and with 98, most of the cells have disappeared. So cells, this again shows that cells that were treated with 46 millijoule per square centimeter, they can recover. They can be seeded again, and they bind the surface, and they divide, etc. But with larger laser fluencies, the result is not so good. So there is a negative evolution of the samples with respect to the control. Samples irradiated with 46 millijoule per square centimeter showed a slight reduction in the proportion of spread cells, as well as a higher proportion of spherical cells not spread on the surface. With the stronger laser energy fluences, namely 66 and 98, the decrease in confluence was more marked, and these cell populations should not be considered as recovered from the laser-activated carbon nanoparticle treatment. Conclusions: the simultaneous generation of hot bubbles and shock waves arising from pulsed laser irradiation of carbon nanoparticles in the vicinity of living cells leads to the observation of uptake or release of cell contents and provides a direct demonstration of the membrane perturbing effects of this photoacoustic event. Parallel investigations using pure lipid vesicles, liposomes, support the idea that the induced membrane lesions are transient and quickly repaired, as deduced from the graded and not all or none mechanism of dye release. Long-term cell survival may not be straightforwardly determined only by assessing cell state through cytometry, even less by only counting necrotic cells. Intracellular delivery methods, which brings membranes under high stress, may provoke subtle cellular alterations that can eventually compromise cell survival. You need a more comprehensive investigation of the consequences of using these methods, measuring cell viability over an extended period of time to elucidate if cells retain their basic functions intact after the process. You can see all the details of this investigation in this paper, where you can find also some videos that you will enjoy looking at. My co-workers, Curro Galisteo here, a professor at the University of Granada, who spent a year visiting us a couple of years ago and was in charge of this research, together with Ingen Monasterio, who is now my PhD student, my brilliant PhD student. And here, David Gil and Miquel Valle from Biogune, the ones who did the electron microscopy, the beautiful electron microscopy pictures that you have seen. Funding from the Spanish Ministry of Economy, Basque government, and the University of the Basque Country. Thank you very much.